My name is Joe Reddy, co-founder and CEO of Partillion Bioscience, and today I'll show you how we're revolutionizing cytometry by enabling the study of cell-to-cell interactions at scale. Partillion is a life science tool startup spun out of UCLA and based in Los Angeles. We are a team of scientists and engineers building the next generation of tools to empower life science researchers. My co-founder and I, having collectively spent decades in the microfluidics field, understood the unique challenges of bringing these technologies to market. While microfluidics tools can be powerful for creating miniature reactions for cells, in practice they require sophisticated, specialized instruments and extensive training. And integrating them into existing workflows has been a major hurdle. Our solution? Nanovials, miniaturized suspendable wells for cells. They create compartments for cells at scale in a parallel fashion without requiring specialized instruments. Instead, we designed them to work seamlessly with tools researchers already have, like flow cytometers. Our nanovial technology is backed by over 9 years of research, published in top journals, and validated by industry peers, winning top innovation and product awards. We have exclusive rights to a comprehensive patent portfolio covering nanovial technology and its accompanying workflows. Previously, we developed assays and kits to enable profiling of individual cells based on secretions, such as antibodies, cytokines, and extracellular vesicles. While single-cell data is powerful, it often lacks environmental context. We can see what a cell is expressing, but not how it is interacting with other cells. Techniques like imaging and spatial transcriptomics provide some environmental context, but they make it difficult to deconvolute specific cell-to-cell interactions. To address this gap, we developed multicell assays that allow researchers to maintain high-resolution data at the single-cell level while also preserving the environmental context, capturing how cells interact in a controlled, scalable way. To illustrate this challenge, imagine running an assay in a standard well plate with a mixture of cells. When a perturbation occurs, you have no way to trace back with certainty which cells or environmental factors caused the effect on your cells of interest. Instead, by isolating pairs of cells in their own wells, or in this case, nanovials, you can now be certain of their environment, enabling precise tracking of how specific cells interact and influence each other. This approach isn't just precise, it's scalable. With nanovials, you can track hundreds of thousands of cell pairings in parallel, enabling high-throughput functional analysis at unprecedented scale. So how does our multicell assays work? Well, nanovials are first modified with cell capture antibodies, which help anchor cells in place during the assay. Cells are then loaded via pipetting and mixing and then incubated to accumulate signal. In this case, secreted antibodies are bound to an antigen-expressing cell and fluorescently labeled. These samples are then flow sorted and sequenced using place-based methods or commercial single-cell sequencing workflow to recover antibody sequences. To optimize our system for these workflows, we engineered our nanovials to be a precise size to enable efficient loading of multiple cell types while maintaining compatibility with standard instruments. We also developed optimized protocols to ensure high-efficiency cell loading. Our capture antibodies are designed for maximum efficiency, allowing us to achieve up to 50% of antibody-secreting cells successfully paired with a target cell. We first validated our multicell antibody discovery workflow using a model hybridoma system. Here, we demonstrated that we could identify and sort out on-target population of cells with over 90% purity using a commercial FACS instrument, and visually confirmed the events using fluorescent microscopy. We further validated the system with a full B-cell antibody discovery workflow demonstrating ability to efficiently recover sequences of antigen-specific antibodies with comparable EC50 values to antibodies used in the clinic. Some of this early work can be found on our preprint on bioRxiv. To recap, the ability to screen libraries of cells based on functional interactions opens up opportunities to enhance and accelerate drug discovery. From enabling efficient discovery of antibodies against difficult targets, to the discovery and optimization of new cell-based therapies. Even more broadly, our multicell assays create the opportunity to better understand the fundamental biology of the hundreds of different cells within our bodies, with the potential to explore hundreds of thousands of different cell-cell interactions. Nanovial multicell assays are unlocking new ways to study cell-to-cell interactions at scale. We're looking for early adopters, collaborators, and strategic partners to help bring this technology to more researchers. If you're interested in learning more, collaborating, or exploring applications, let's connect.